In this lesson, we're going to shift away from DNA replication and attempt to understand the connection between genes and proteins. Of course, from your grade 11 backgrounds, you already have learned how genotypes can actually result in different phenotypes. Remember, the genotype is what's found in the DNA, and the phenotype is really the trait that's exhibited. So Gregor Mendel's P's come to mind. Well, in the early 1900s, a scientist by the name of Garrett actually studied a disease, alkaptonuria, and this disease was interesting because it turned the patient's urine black. And after studying numerous patients, Garrett figured out that many patients with this disease all kind of had the black urine, and that is a phenotype. And he started wondering if there was a gene that was in charge of causing this change in the protein. And of course, that was the very first time that the link between genes and proteins was made, and the whole idea of inheritance kind of became a reality. Later, research actually confirmed that this disease is caused by a genetic mutation that normally codes for an enzyme that breaks down the amino acid tyrosine. So Garrett's research in the early 1900s later became proven that there are genes that code for proteins. So the whole concept of genes causing different traits became a reality. The genotype is, of course, what is in your DNA right here, and the phenotype is the trait itself. So this is kind of the setup that Garrett started in the early 1900s, and more studies were carried out around the 1940s that I'll talk about in a minute here. This led to a very important concept, which was called the one gene, one polypeptide hypothesis. In a nutshell, a lot of people were starting to believe, including Beadle and Tatum in 1940, that each one of our genes codes for a polypeptide, which of course is another word for a protein. And Beadle and Tatum studied the red bread mold fungus, which of course, if you have moldy bread, this is probably the one that you've seen or even picked off a moldy piece of bread. Not that exciting to us, but to Beadle and Tatum, it was pretty important. So let's take a look at their study. The metabolic pathway for bread mold, or Neurospora crassa, involves 4 metabolites. Metabolite A is sort of the beginning, and metabolite D is the ending. In this case, to make that bread mold, you need these 2 intermediates, what we call B and C. Each of those steps has to be catalyzed by a specific enzyme. And I'm not really worried about what we call those metabolites or even what we call the enzymes. The bottom line is they studied this pathway, which involved 3 steps. A going to D through B and C. There's a picture of the bread mold on a petri dish. Not that exciting to us, but certainly exciting to them. And of course, it's formed when A is converted to D with all those enzymes present. So here's their experiment. Bottom line is they took the bread mold spores and irradiated them with X-rays. Then they took that bread mold that had been irradiated and cultured it on petri dishes, and the enriched medium just basically means that all 4 metabolites, A, B, C, and D, were present. The enzymes were present, and that allowed the bread mold to grow even though they had been irradiated. Interestingly enough, when they put the bread mold on minimal medium, which means there were no metabolites or enzymes there, the bread mold was not able to grow. And normally bread mold would grow with minimal medium. So this kind of gave those 2 scientists the idea that something from the X-ray had changed the DNA. Well, further studies showed that when they took bread mold from the enriched medium and recultured it onto a blank petri dish that only contained metabolite A, the mold did not grow. Furthermore, when they took the enriched medium mold and put that into a petri dish with metabolite B, nothing happened there as well. So that kind of implied to them that something was going on in the latter part of this transition from A, B, C, and D that clearly did not allow this bread mold to grow. Interestingly enough, when they included the third metabolite, C, that bread mold did grow. And obviously, when they included metabolite D, the bread mold grew there as well, or grew there, I should say. So a number of conclusions could be made from this data. Number one, Beadle and Tatum determined that normally bread mold will grow on minimal medium. It doesn't take a lot for mold to grow. But when you expose the bread mold spores to an X-ray, obviously the DNA was altered. And of course, we know now those are known as mutations. And that caused the mold not to grow on minimal medium, but it did grow on enriched medium. More specifically, this bread mold needed only metabolite C or D to grow on the minimal medium. So the final conclusion that Beadle and Tatum came up with was simple. Apparently, this bread mold needed enzyme 2 and could not change B to C. It could change C to D. And that sort of was the first experiment that demonstrated this one gene, one enzyme hypothesis. Without that second enzyme and without the ability to convert B to C, the bread mold could not grow with the medium. So this gave rise, of course, to the whole concept of converting DNA to RNA to protein. So in a way, we've kind of circled back to Francis and Crick and the good old central dogma of biology. We have understood at this point how DNA replicates. It's semi-conservative, and we know that an exact copy has to be made for DNA to be functional. We talked a little bit about mutations. And of course, DNA replication occurs in the nucleus of the cell. Our next step is to figure out the transfer of DNA into RNA. RNA is really the message that gets carried from the nucleus to the cytoplasm, and finally, that RNA has to be translated in the cytoplasm to make protein. So the Beadle and Tatum experiment kind of showed that DNA in an indirect route controls the protein that gets exhibited or produced. We have to look at the processes that occur between the replication and the protein. And of course, that's the production of RNA and finally the production of protein. Hopefully this helps. Our next step is to focus a little bit more on RNA and the genetic code.